kick off the recording. But yeah, this is our bi-weekly interest group meeting. So if you're in the Boston area and you want to come by, you're always welcome to come. It's just an informal sort of meeting where we talk about work that we're doing, and just try to share tips and tricks and everything that we're doing. And we sometimes broadcast it depending on how prepared people feel. I didn't feel very prepared, but I like to broadcast it and make everyone available. So I know what's happening. So cool. I got a ton of slides, and I'll just kind of jam through. If people have questions or want to talk about things more, just let me know. I hate talking for an hour straight. It's very painful. So please interrupt me if I'm going on and on. What I'm going to talk about is stuff we've been doing with bcbio and Common Workflow Language to develop interoperable, collaborative, multi-platform variant calling. That's pretty cool. And if I'm going to record this and I'll post it as well. So if you want to share with anyone, that's absolutely cool. The slides and everything will be at j.mp bcbio links, which is linked to the bcbio presentation pages, or you can remember bcb.io. So they'll have any URLs or anything you might be interested in. So what I want to talk about is the barriers to building analysis pipelines, like what makes it hard to build an analysis pipeline, and then talk about bcbio, which is the analysis platform that we develop, and how I think open source community development really helps build quality pipelines. And then I'm going to talk about sort of the next level is how can we integrate bcbio in other places, and specifically using the Common Workflow Language, workflow description language, CWL and WIDL, which are considered like assembly languages for workflows. And then to sort of demonstrate how this is useful, talk through a lot of practical CWL things we can do with bcbio, so running on HPC, running on GNexus, running on Avatos, running on certain bridges, and then talk about how hopefully in the future we can automate this and full multi-platform testing and validation. New people up there. So, but anyways, yeah. And I think the three takeaways I hopefully want you to get from this are, one, that the most important thing about science is being able to collaborate and reuse other people's things. And so, one thing we should be striving for in Linux is how we can best collaborate, let people reuse the things we're doing and work together. And I think to do that, we really need these interoperable workflow abstractions. And if we do all these cool things, we can build better things together. And so hopefully this is demonstrating how that's happening. And yeah, if you remember last year, I've talked a lot about this sort of idea of Common Workflow Language, and sort of had some proof of concept. And this is really productionized and ready to go. So, hopefully you'll see sort of some progress from last year and not the exact same slides. So to kind of motivate, let's talk about building variant calling pipelines. So this is a GATK best practices, which is re-implemented in tons of places all over the place, in shell scripts and in specialized workflow. And everyone takes sort of what GATK says and it's an awesome pipeline, does variant calling, does a great job. And what I talk about is how we can make that pipeline so we don't all have to rebuild this pipeline ourselves. We can run it in our environments. Because you want to build, what you want to do, this is my energy in and energy out diagram, to think about like how the sort of effort you put into things and what you get out of it. So ideally, you put in this effort and you'd like to get out science, right? The E out is interesting science, helping people do things. And the loss is all the time you spend making the workflow work, bugging problems, tools, figuring everything out. And we want to try to maximize the science, minimize the amount of time we spend fucking around with the different tools and workflows that work. And so much effort, and there is so much loss, is that there's various, and I'll talk through sort of each of these in more detail throughout the talk, but tools are changing all the time. You have to support a whole ton of different features and platforms, which creates a huge support burden as you do that. And then you have to validate and make sure things work every time you're changing. And so these are all this work that you do that's ancillary to the things that you like to learn. And I think like the best way to work together and avoid effort is by building open source communities. And it's not a surprise to anyone. But if you're interested in this kind of thing, this is just a little shout out. Next year in Portland in June, there's the yearly bioinformatics open source conference with Galaxy Community Conference to create this big bioinformatics community conference. The idea being this is a place for people who are interested in open source and community development and bioinformatics. It's going to be awesome. We have a lot of flexibility. There's training days, there's coding days where we work together after the conference. So if anyone's interested in kind of out here, you're very welcome next summer in Portland. And what I talk about, but the actual practical like open source thing I'm talking about today is this is sort of the high-level cartoon of what it does, but basically it takes sequencing samples and a configuration and integrates a whole bunch of tools, provides scaling and resiliency, and then on the other side you get out things like variations, RNA-seq outputs, quality control outputs, and into downstream analysis tools so you can do other cool things. And this is all open source and freely available. So this is the GitHub link if you're interested in that. And today I'll talk a little about variant calling sort of as a science side of thing, but we actually do a bunch of different variant calling, and I think Miranda, you might be having a couple weeks. Small RNA, we do RNA-seq analysis, single cell, ChIP-seq, all kinds of, there's all kinds of pipelines in there. So to sort of make it practical, what we do is try to codify these kind of, variant calling or other analysis methods and make them usable in an easy to use way. And so we made a pipeline. So what? There's a lot of different pipelines. And this big, I like this big paragraph describing sort of a pipeline and sort of all the different other pipelines and why you have to build a new pipeline. One of the things that we're very good at in bioinformatics is building pipelines, and one thing that's very difficult is being able to maintain this. But I think this is an awesome definition of sustainability from software carpentry. It's that a piece of software can be sustained if people are using it, fixing it, and improving it rather than replacing it. And I think one of the things that we are bad at in science is being able to sustain and existing tools. We kind of keep reinventing the same thing over and over with slight improvements. And this talk is sort of about how can we doing that. And the reason that it's so hard to maintain and keep things up to date is that things change all the time. So this is GATK best practices, which every time I update this, I get a new thing, I think. But so bcbio has been around for a long time. Originally, it was command and just grab a seat. bcbio has been around for a long time. The initial version of this was when I was working at MD. We wanted to run the GATK best practice pipeline, and there wasn't really a way to do it. And so we implemented the whole genome deep coverage version 1. And since then, there's a version 2, there was version 3, then there was another one, and then there was GATK 3, and upcoming in January 9th next year, set your calendars, GATK 4 will be released, so there's another GATK, and so that's 6 different revisions of the pipeline tools. You have to validate, update, and so I don't know. The sad thing is, if we're all developing this ourselves every year or so, whenever this changes and improves, we're updating all these tools ourselves everywhere, redoing the same thing over and over, which is a shame. And the other flip side of this is that you want to have your pipeline, you want it to run in a ton of different places and do a ton of different things. So this side is intentionally not able to be seen. What it is a big list from the next book, which is a really cool paper and a really cool pipeline system. talking about the things that Nextflow does and other workflow systems does, and yes, no, do they support any of these things? And if you're looking to build a new pipeline or make it run, you suddenly start thinking, well, which of these, I don't know, 30 different places and things that people want to have do I support and work? And so it's a ton of work to do and keep up to date. Yeah.
Can you qualify them? Because there are people that if you can mute it.
Oh,
Somebody. Oh.
sure, I can try to mute them. Manage. Yeah, I can mute. It should be. Where did it just go? Mute. Yeah. Am I the only one who has permission to do this? Okay, I think everyone is muted now.
Now it's better. I think I got it.
Cool. Okay. Fabulous. Fabulous. Thanks for interrupting me. I was talking too much. Cool. So I think the most important thing about being able to sustain and support something is being able to build a community around it. So this is sort of demonstration that bcbio has been around for a long time, since, I don't know, 2010, and the graph on the top is the commit graph over time, and you can see that we've been doing things for that whole time, improving, updating, and we've also been supporting bcbio. So there's these slides are actually a little outdated because there's like 2,000 issues or something now. Issues pile up quickly, but we have an active GitHub. We answer everyone. We try to help people. And so this is the kind of like effort that is a ton of work. It's sort of ancillary to the biology, but important to help people get things done that we should be working on together as a community. And to build a pipeline or a set of things that people want to run on, you have to have these really lofty goals, because people run things everywhere in many different places. So you want to run things on local machines. You want to run things on clusters, on different clouds. You want to support people who are working in clinical environments. You want to have an interface for researchers who, maybe don't feel comfortable on the command line. You'd like to integrate with LIMS systems, so things that are output directly into the pipeline and run. And I mean, ideally, you'd like it to be accessible to the general public. So, a person who gets their genome sequenced at Veritas for $1,000 and wants to reanalyze it can use your tools to do the same thing that you're doing. The problem with this is that we're moving data all over the place, and we're not. So that one makes it hard to support all these places. So this is a kind of we haven't been improving that much slides. This is a tweet I dug around from DNAxis 2013, where they talk about you don't got to move data outside the cloud. You can do the analysis right there. And some other, from Stewart in 2014. This big change coming in genomics. We're not going to move data around. We're not going to download it. We're going to move the code to the data. We're going to do it that way. And I talked about this at ISMB, and like the day before, there was a conference when Bernie was talking, and he was saying, "Oh yeah, we're moving it. We got this big problem. We got to start moving the analysis for the data, right?" And then I looked last night, and there was another tweet from Aaron from Genome Informatics is going on, and people are talking about, don't move the data, move the analysis data. That's the only way we're going to scale it. So everyone knows the problem. We've been talking about it for at least 4 years on Twitter, whatever I could find on Twitter, but much longer. And why are we transferring data around instead of moving analysis to data? And the reason, I think, is that it's just a lot of work to set up and configure analysis. So once you have it running, you saw all those platforms that are listed, and once you have your SGE custom analysis set up on your system and it runs, you're happy, it's cool, everything's good, and then you want to move somewhere else. You want to move to Amazon, or you want to move to a different scheduler. You have to port that whole analysis to the new environment, and there's a huge overhead to doing that, and it's a big pain. And one thing that starts to make that easier are workflow systems. So I'm going to highlight a few, I think, awesome workflow systems that try to tackle this issue. Nextflow is one that that's really awesome. That works in a bunch of different environments. Has a domain-specific language that you use to define your workflow. It sort of helps you start to move take one thing in one place and then run it in another. Another awesome workflow system I talked a little about at the conference is Galaxy. This is, they have a huge focus on reproducibility. They have an awesome web interface that people can use to run analyses. They have they contribute a ton to all these open community projects that are building up ways to build analyses. And it helps you take your and it runs on top of multiple platforms. So you can run Amazon, you can run it locally in a cluster. There's systems like SnakeMake, which is another great system which runs on clusters, which runs locally, parallelizes things for you, has its own sort of domain-specific language based on Python that you can use to run workflows. And what I'm trying to get at is there's many great workflow systems. And so this is a wiki page from the Common Workflow Language with a list of existing workflow systems. And I didn't fill the whole page because I just did the end because there's 181 different workflow systems that they have listed. And many of these are new. So there was just this cool one out from Grail Bio called Reflow, which is a nice looking workflow system. There's a ton of awesome work going into building these analysis pipelines. And the problem is we'll never agree on one system. This is the same as trying to agree on a programming language or trying to agree on something else, right? Like each thing has a different advantage and disadvantage. People use them based on their familiarity or what they're taught, or there's just personal preference, right? Scientists are, and programmers, you get both of them together, right? And you'll have hundreds of different opinions, and you'll never get people to agree. So hopefully we can agree that we're never going to agree. So that's an issue, right? We can't easily share workflows. If you're built into, if your groups, you all use Nextflow or your groups all use Galaxy, that's awesome, right? You can use that workflow system. You can pass things back and forth. You can share the work you're doing. Everything works perfectly. But if you want to share externally, it's a huge barrier. If someone's using Galaxy and I'm using Nextflow and they say, hey, I wrote this great workflow system in Galaxy, why don't you use this? You have to then spin up Galaxy locally, run it, learn all about configuring it and making it work. It creates a big barrier. And people, in the end, people don't do it, right? If they're not built it, they're already into it, they just rewrite it on their own thing. And ideally, you'd be able to mix and match components, right? So I can say, hey, someone wrote a great variant calling workflow on Galaxy. I'd like to take that and plug it into my downstream analysis and run it all on my platform and workflow. We can't do any of those things, or it's really hard to do any of those things right now, so we don't do them. So the question is sort of how can we do better? What can we do with all these workflow languages to do better? And the answer, I think, is to build abstractions, better abstractions that make things interoperate. And the abstraction I'm going to talk about today is a sort of a standardized workflow language. I'll talk a little about Common Workflow Language and WDL. And the work we've done so far is with Common Workflow Language. And what it is a standard way of defining how your pipeline runs. So if you look underneath any workflow system, there is some way of defining the workflow, either explicit or implicit. So like graphical interfaces like Galaxy, they have a workflow system underneath, but it's not exposed to you. There's things like Nextflow, they expose their DSL to you and it builds it. And then that's compiled into some internal thing, then gets run. And so what Common Workflow Language tries to do is to be like sort of the assembly language of that thing that the workflow language is compiled to. So it defines, this is the sort of stuff it defines, like you have a big workflow, you have this sub workflow within it that's doing something, and you have a bunch of steps that are running tools, doing other things. And these are all the things that you're defining essentially in however you're writing your workflow, even if you're writing it in Bash or whatever, you're defining these somehow, but this makes it explicit. And And with CWL, I want to sort of emphasize is sort of one way of thinking about this problem and sort of with one representation. The goal really is to have, let's compile to one set of things that all work together. And another great initiative from the Broad Institute here is the workflow description language, or WDL, which is this open model. If you go to openwdl.org, they have sort of an open discussion standard, the same as Common Workflow Language, and that's an alternative sort of front end to defining workflows with the aim of being portable. So I think my sort of unhidden goal is that WDL and CWL will compile to the same thing and will represent the same things, and you'll be able to interoperate between them. And those present you with sort of a different way of writing and representing your workflow. So give workflow authors, workflow tool authors, better ways, easier ways to plug into these tools. Yes.
If that means, so you've got CWL in the middle.
Yep.
Is somebody else out there also trying to do the same thing?
I don't think so. It's a thankless task. So like, I don't think anyone else is really trying to do that. I think those are the two sort of, and the difference really is sort of where the emphasis has been so far. CWL, the emphasis has been on building a language that a lot of platforms can support. And WDL, a lot of the emphasis has been on like making it easy to write interface that pro scripters, pro loop and script things like to use. But I think they're coming together now in the sense like people are actually using them, and people who use CWL say, hey, it'd be nice if we had a nicer DSL to write this. People who like WDL are saying, hey, it would be cool if this ran in multiple places besides Cromwell. And so, yeah, I think, I don't know, like hopefully no one else wants to do anything, because like it's a Herculean amount of effort as are all these workflow systems, and these have been sort of in progress for years and kind of moving. So I hope there's no more. We'll just, whoever wants to do it can kind of come together. That would be awesome. Much like CWL and WDL are trying to come together. Cool. So I'm going to try to demonstrate why a workflow abstraction is great and helps you do really awesome things by showing you how we're using this in bcbio. So I think the important thing that does is let you integrate with multiple platforms. So I'm going to talk about how right now with bcbio, you can take it, you can run it locally using Ravix Bunny Runner, you can run it on HPC using the Flow Runner, you can run on different cloud platforms using 3 different providers, Arvados, DNA Nexus, and 7 Bridges, which also runs the Cancer Genomics Cloud. And the reason that this is also a ton of work for bcbio to support CWL, and the reason we're doing this is that we don't want to maintain bcbio specific infrastructure. I don't want bcbio to have a AWS runner. I don't want it to have a HPC runner. I would like it to work on these other great runners and great workflow systems that already exist, and really just focus on the hard biological problems, making a good variant calling workflow, making a good RNA-seq workflow, integrating lots of tools. There's so much work to do on just that. You don't need to also build out infrastructure as well. So that's why we're trying to do that. And sort of, I think the unique sort of goals with CWL is that you can have the same production environment in multiple places and switch back and forth. So you have a local HPC that you run on, and you're running all your things at Slurm, and you're happy there. And then you get this big analysis project, and I want to run it. And then you can take the exact same thing you're running there and put it onto DNA Nexus and run in DNA Nexus or some Bridges or Autos. Or you could, if you want to run direct on the cloud, you could take the exact same thing and run there, on top of cloud provider. And the idea is to have a coordinated release and update process, so you can know the exact version of a workflow you're running, the exact set of tools and containers that you have, and the same reference data. And once you sort of have those 3 things, you're running the exact same thing. And that's a sort of a really hard problem to do right now. But what it does is start to enable these connections, and then people who would like to run on DNA Nexus and like your workflow that you wrote, that you're running locally on your HPC, can take parts of that workflow and they can run it on DNA Nexus. And you do start to get all these communities working together and building things together. And so the rest, or the next part of the talk is really trying to like demonstrate that I can actually do this, or we can actually do this by sort of walking through the examples and showing how you go back and forth between different places. So it's kind of walking through how you run bcbio. So you start with this high-level configuration file, you generate CWL, and then you run it on the platform. And the platform, or whatever you're running it on, has the CWL and the tools in some way. That can either be, right now, a Docker container or a local bcbio installation if you're running locally on a shared file system like the HPC environment. And then you have the genome data. So what I'm going to kind of try to show is that, so genome data is there somewhere, either locally or on the system. And I'm going to show that you, switching between these platforms, you generate CWL in almost the exact same way from the same configuration file, and you run it in almost the exact same way in all these places. So you really are interoperating back and forth. And the glue that puts this together is called bcbio VM. So the idea is, if it's confusing, is that bcbio itself, or as it has been, is meant to be sort of the place where all the workflows, where the actual tools get run and things get described, and there's biological insight. And bcbio VM is meant to be sort of the glue around that integrates with external tools, that makes it easier to run different tools and that kind of stuff. So trying to kind of separate what's the workflow from what's the glue that connects these workflows together. And so if you want to try this out, everything's in Bioconda. So, which is, if you don't know Bioconda, you should absolutely use Bioconda for installing your tools. It's an awesome set of packages. And they have, I forget how many packages, thousands, right? Of packages, 3,000 packages, something ridiculous, that the whole community of people is maintaining and keeping up to date. And so you can just run this one command. You get bcbio, you get bcbio VM, you get all the different runners that I'm talking about here installed. Just a bunch of magic all in one place, just by running that, and it's works across a bunch of different platforms. Not all on Mac yet, but that would be awesome if it also installed on Mac. So cool. Once you've done that, then we're into actually running bcbio. And so if you've run bcbio before, this is probably familiar, because it's the exact same thing you use to run it as it is right now without CWL. It's just a simple sort of YAML markup that you describe what you want to run. So this example is you're aligning with BWA, you're doing GHK recalibration, you're variant calling with haplotype caller. This uses GHK4, and you're doing joint calling, so you want GBCS, and it's over these exome, the exome region plus chromosome 20. This is sort of an example validation workflow we're putting together. I'll talk more about later for the GA4GH Dream execution challenge, and you're running on build 38. So it's really sort of a high-level description of what you want to run, right? And then what happens with that is then you take that's sort of hey, this is what I want to run, and then you need the way to describe, hey, here are my samples that I want to run. And so, there's it's just a CSV file of sort of sample and description, and the batch, you want to recall things together, and then this is running validation, so validation sets associated with each of those. So this example is just it's a genome in a bottle sample, so NA12878, 24385, and 24631, which are a Caucasian sample, a Ashkenazi Jewish sample, and a Chinese trio sample. So like a diverse set of genomes that you can run validations on. And then the final thing you have to do is define what you want to run, where things are at, tell it where things are at. So there's a second configuration file this is the local setup, so either on a local or shared file system, where it's describing where are the reference genomes, and in this example, there's in this biodata collections, and then where are your inputs that you defined in that CSV file, so it knows where to look for those. And then the type of resources you want to use for running this. So, I'd like to use, when I'm running BWA, I'd like to be using 8 cores with 4 gigs or 3.5 gigs of RAM per core. And these are the options for if I'm running a Java programming or something. So this would be good for like 8 core boxes with 4 gigs of memory per core. Sort of telling it what the resources are. So those are the 3 things. And from there, it's 2 steps to generate a common workflow language description. So the first thing there is just like I give a project name, it's the genome join calling. And then you run this template command, which takes that CSV file I described and the template of what you want to run, and that generates a full configuration. So, for each of the samples, I think there's 3 samples in that example, right? So it takes a template and generates 3 files for each of those. And then the second step takes that output, which is a single file, and converts it into common workflow language. So it creates this directory of a workflow description in common workflow language. And then once you have that, you can just point at that directory. It's called the project name workflow, and run it. And that's it. So this is bcbio VM, CWL run, Bunny, and that workflow. And that takes the Bunny runner, which is from the 7 Bridges folks, a pretty cool open source runner that runs multi-core on single machine, and will run the analysis, split things up, do multiple cores, do all the parallelization, handle everything for you, running right there. And the cool thing is then if you want to take that exact same common workflow language and that you output, and so you were running, say, on a cluster on a single machine, and now you're saying, hey, I want to scale up on this machine and run on Slurm, you can take that exact same common workflow language, so there's no recompiling or anything, it's the same file system, right? And then you can run with Toil, so a totally separate runner. Bunny's written in Java, this one's written in Python, they share no code at all, they're completely separate. You can do bcbio VM, CWL run, Toil, and now you're running on Toil. And so this has some other, you can also change things around a little bit. So this is like this running without containers. So say running an HP system where you don't have Docker, and it's submitting to Slurm. So you can define some things happening in Slurm, right? Like I want to run the short queue and give 12-hour timeouts. But the cool thing that just happened, hopefully is cool to people besides just me, is that you switch between 2 workflows with the exact same thing, and it's almost no work to do those 2 things right there. You learn the one way of doing it, it's almost exactly the same. And so what I like to show is how then you can go, so that's on local machine, right? I like to show how you can do this when you go to cloud or on different vendors and things. So this is Arvados, which is a nice workflow system from Veritas and Pureverse, and they're one of the folks that were first involved with writing the CWL spec. So they've been sort of all CWL all the time. And so what I want to kind of show is this is the description, which is very similar to the local description we had earlier, where before we were defining local and the reference to the inputs. Here, the reference to the inputs are these hashes, which are references to an Arvados. So Arvados has this full idea of everything being you have full provenance over all your data and everything, and so they have these unique reference hashes. Most of the workflow systems have the same thing, where you just knowing that GUID, you can then look up exactly what files and everything are. And so those hashes, the reference is a reference collection with all the bcbio stuff, and the input is another collection with all the input files. And then the resources is the exact same description you ran earlier. So I run on each of the stuff with 8 cores and 3.5 gigs of memory. And then the cool thing is that to run that, you're doing the exact same thing you did before. So the template in CWL generation steps are exactly the same as before. Now, instead of pointing at that local, YAML file that described your local environment, you're pointing at that Arvados one I just showed. And bcbio will go, uses the Arvados API, it looks up those reference hashes, it finds the files, it gets the references to all the files, and sticks them into the CWL that it generates. And then when you want to run it on Arvados, it's exactly the same thing as before. So bcbio CWL run Arvados, and then the workflow that you just generated, and then specific options for Arvados. So project that you want to run it in, that you're sticking it in. And that then submits the job to Arvados, runs it there. And so using the exact same system that you set up, that you use, now you're running on a cloud system that's completely separate. And this is also, and then this also works on 7 bridges in the Against Genomic Cloud as well. And you'll notice it's almost the exact same thing. So instead of defining local or Arvados, you define 7 bridges genomics, and this is the project I'm running from, and this is where the reference data is, and then the resources are exactly the same. And you do the exact same process. So you generate the CWL, it looks it up from 7 bridges using the API. You then submit it, and it runs there. And the cool thing, I don't want to belabor showing the exact same command lines over and over again is kind of boring, right? But this is a completely separate setup, right? So they have this. This is a bucket on the Against Genomics file with the HE38 metadata. It's all there, loaded. You can pull from it. We can share that resource amongst anyone using Against Genomics Cloud. It really starts to enable these cool things that you can do on these platforms. And so this is DNA Nexus, which is almost identical again, right? So you have your project, you have the reference, where the reference material is, where the reference stuff is, so where the genome is. You have the inputs, and then you have the resources again, and this does the same thing. So you run, and run the templating, you run the CWL, and it looks up, uses the API, looks up on DNA Nexus, finds the references to all the files, sticks them into the right place to be, and then you can run it. And I think one of the cool things is, if you know about CWL, you might think I'm cheating, showing you 7 bridges in Arvados, because they're sort of the founders of CWL. I'm one of the people that have really bought into it from the beginning. But DNA Nexus, they have their own workflow language. They don't use CWL internally at all. And what you can do, which is really cool, is when to running it, you're taking the CWL that gets generated, which is the exact same as the other one, the other platforms, just with different references to where the files are, right? And now you're compiling it into the DNA Nexus workflow language. So you're really taking that CWL and using it as this intermediate to write to DNA Nexus. And so there's this 2-step process to submit, which I thought was worth showing to show we could automate this as well. So it's a bcbio command, but I thought it's worth showing just to show the cool things that are happening underneath, is that it's compiling the workflow into DNA Nexus apps and their workflow language, and then it's submitting and running the workflow in their system. And they have an open source tool called DXCWL, which does all that compiling. And once again, you're getting cool things. I'm just trying to show kind of from every platform different cool things you can do, but you get cool things like monitoring. So this is a workflow that ran that you can see the whole Gantt chart kind of the breakdown of the process. You can see how long each of the steps took. You can drill into each of the steps and see what took a long time. And I mean, this is really cool stuff that you don't, sort of insight you don't get if you run an HPC, unless you have things set up to somehow generate these. This is sort of built in, and immediately you can see, hey, this step, whatever is, the QC, there's some steps here that need some optimization for sure. There's like this, these are converting to records and they take a long time, and I'm not sure why. It's all new stuff that just got ran this week, but it's really easy to see what takes a long time, what's taking your process. You can see what's happening in parallel. So, up here you're doing alignment, and then once you're done with alignment, then you can do QC on one part, and you can also do on other machines. You can run variant calling and joint calling. This is a full, the GHK4 joint calling pipeline. But you get these really cool things. So I guess what I've been trying to show with that is that this is really running in many places in a standardized way, doing lots of cool things. And we're actually making real progress in terms of using common workflow language to do things that I think are very difficult to do with a sort of just a single workflow system if we try to focus on a single workflow system. And so on the topic for a while, I might be losing anyone. Okay. I don't want to close yet. So the last thing I want to talk about is sort of how we can make this automated and validated. So this is awesome, and it's a lot of things running in a lot of places, but I talked a lot about support burden before. The more places we get it running in, the more we need to test and make sure it continues to work there, the more we have to validate every time we update things that we don't break things. It creates potentially a lot of work. And so how can we try to share that work and work on it as a community? And so I think the way to do this is through validation. bcbio is really big into having validation. I think of this as integration tests for pipelines. So you have lots of unit tests and things run, but you need a way to make sure that you're actually producing the biology that you want. And it also lets you do really cool things like do algorithm comparisons in an unbiased way. I don't care if GTK4 or Freebase or Strelka is better. I don't have any, I just want to run the best tool. And so this is a platform to let you do that. And then also a baseline to improve the method. So if some tool is doing worse, hey, why is it doing worse? Could we improve it? And like I mentioned, a way to test on platforms. So now that we suddenly have things working on 3 different platforms and we make changes and release a new version, it would be awesome if we can run these validation tests on those 3 platforms, make sure that nothing changed, didn't break anything, everything runs good, so that people are picking up and taking it, don't have to find those bugs themselves. And so this is all based around having reference materials and standards. And so the people that have been pushing this forward are the Genome and Bio Consortium, the Global Alliance for Genomics and Health, and all the different dream challenges on the Synapse platform. They're really, they're an integrated set of communities that are building both reference materials and sort of the standards and methods and tooling to help make these sort of things possible. And what you get out sort of from bcbio, I wanted to show some biology. I felt like I was talking about all things, so I'm going to show a little about biology. So I'm going to show a validation graph on the next page, and this explains what it is. Just an example one. bcbio spits out these comparisons against reference materials. So on the next page, I'll show one against Genome and Bottle, and but this sort of breaks down what happens. So on the top are SNPs, this is variant calling, on the bottom are indels, and then on the left side is sensitivity, and on the right side is precision. And so you get an immediate sense of, this is just an example with 3 different methods of, how the methods compare in terms of sensitivity and how they compare in terms of precision. And the only, I think, tricky part is that lower values are better. This is because it's hard to plot when you're talking about 99% concordance versus 99.7% concordance. If you show that on a 0 to 100 scale, they look exactly the same if you show 2 bar charts, right? And if you cut off the bar chart at some arbitrary point, you're not supposed to do that. So these are basically 1 minus those rates of sensitivity and precision, so that you don't have to do that. And so just to show that example I showed before that we were running, where we had the 3 genome and bio samples, the 12878, the 24385, and the 24631. And I didn't talk about it, but they are actually 3 different technologies. So it's NovaSeq X10 and HiSeq 2500, and then the X10 has the added caveat that it has 10x tags on it, so that creates some problems with false positives over here. But it was kind of meant to show, this is in those validation pipelines are meant to show, like, hey, you can rate these things that compare all these things, you can run them in all these different platforms. And then you can start to look at, sensitivity and specificity across the different platforms and see how things compare. And not any big takeaways from this other than, like, hey, things all look good. These are all GTK. This is all JTK4 joint calling on build 38. Ideally, what we'll do is then have this and have comparisons with other tools like Strelka. And if you go to this link at the bottom, there's a whole set of these different validations with comparisons to different tools like Freebase and Strelka, and showing how in GTK3, and just showing things are working pretty good with GTK4. And I'm excited about it because it's open source, and we can include it in bcbio again. But you can see that this is, we've been talking, as I mentioned, it's a ton of work, right? So am I going to run all these? This is one validation. We want to run semantic validations. We want to run structure variant validations. We want to run RNA-seq validations. We want to run small RNA validations. Are we going to run these 10 different validations against 7 different platforms every time we change something? That's a pain, right? I mean, I'm not going to be pressing 70 buttons every time. I don't like pressing one button, so I'm not going to press 70, that's for sure. So we need some kind of continuous integration process that makes this possible. Automate the testing, and then you can start doing like, hey, here's a new workflow definition. Can I plug it in? Here's some new tools and algorithms. How do they look at all these places and make a transparent process? And the group that's promoting for this is the GA4GH Workflow Execution Challenge. This is ongoing right now. If you go to this website, there's a set of WDL and CWL workflows that, including a bcbio one, that people are running across multiple platforms. So there's been a bunch of different platforms that ran these and submitted back their results. There's a validation checker that checks the outputs. So like in this case, the sensitivity and specificity against what you expect. Make sure that things run equivalently across all those platforms. And this is, I think, a first step in terms of getting things running in multiple places, demonstrating the work, demonstrating that common workflow language will really run in multiple places. And the next step is trying to automate this and make it that continuous integration process. So GA4GH, in addition to CWL and WDL, have a whole slew of acronyms with different standards that span sort of the whole set of executing things. And the sort of next step up is this WES, Workflow Execution Service. And what you can do is if
Not to be confused with Cole, actually.
Exactly, yeah, or some person named Wes, right? You know what I mean? Like, exactly. There's a whole different set, but yeah. So you can just say Workflow Execution Service. I think that's the right acronym. May have messed it up. Or Workflow Execution Schemas maybe is the right. So I messed it up. But what it is the shared API for running workflows. So it's a set of REST calls. If you implement those on your platform, it can push to you, and it can run your workflow. And so this is something that's not existing yet or is working to exist, right? So if this kind of thing sounds cool, we are welcome contributors. I think this would be an awesome sort of, this is basically like a Travis CI. The idea is like a Travis CI or something like that for workflows, biological workflows, automating the process of that. Whew. So no one thought I could. Oliver didn't think I'd get through all the slides. He said I had too many, so I did get through. I'm at the end. So I just want to reiterate sort of the things that I thought were the important takeaways. I think that doing science means collaborating and reusing things. And the way we do things now is not optimal for being able to reuse it, reuse things. We can run things and get workflows, but it's very hard to translate between those. And if we can build abstractions and work around abstractions like common workflow language and WDL, we can interoperate. And this, hopefully I've demonstrated that, hey, we're doing this in the context of bcbio, sort of like showing that what is possible doing using all these things together. And so, that was sort of the high level takeaways, and then all the details are just, if you're trying to build an analysis workflow system yourself, it's really hard. The tools are changing all the time. You have to support a ton of different features. You have to work across multiple platforms, and you have to validate. It's a lot of work. And we should be doing it together. And bcbio is the thing that we're trying to say, hey, this is the way we're doing it together. Common workflow language and WDL are the ways that the community is saying, hey, we want to run our workflows in multiple places. Here's the definitions we can use to do that. I showed how bcbio is using CWL in a practical way on HPC, gain and nexus, or how some bridges to run things with the standard inputs, standard way of running them, across lots of different environments. And then I talked about sort of hopefully, I feel like I always give the exact same talk, but it's progressing, right, every year. So if I give the exact same talk next year, the GA4J stuff, the automated multi-platform validation, you'd be like, hey, look, now we have this awesome stuff. It works on these 8 different systems. Here's the validation tests that run every month that show, compare these 5 different tools and show you how things are progressing over time, or not regressing over time. And yeah, that's sort of hopefully the future of doing things. And if any of it interests you, it's you can remember the main website for bcbio is bcb.io. All this is open source, good stuff. So everyone is welcome to contribute. Cool. So I talked the whole thing. I have like a couple minutes left. I'm happy to answer questions from here or from people who are, I don't know if everyone fell asleep online, but half the people are asleep here, so it's good. Cool. Thanks, guys.
So, Brad, great talk. This is Ravi. I had 2 questions. One, when you showed the batched execution of the workflows, two important things. One is, how is the data locality? How is data getting to the analysis steps? Is it assuming that there is a shared file system, and how the analysis tools are finding data is by co-location of data? The second question I had was, how is the parallelism being handled? Is it in the code to kind of split it by the sample? Do you do more by chromosome level or other types of parallelism within the analysis? Sure.
Yeah. Definitely for the batch system, you're kind of at that point you're assuming sort of a shared file system. I think like Arvados and DNX and Bridges, that's sort of more the model we would like to move to, is where you have some stable hash reference to a location, and then you look that up, and then you can have sort of other ways of pulling the data back and forth. And we have some of that, like with Amazon S3, but I don't know, you could maybe use Swift or something else locally for another object store. But yeah, like mostly the most common case we support is a shared file system in HPC. So yeah, it is assuming that.
So one of the things that we worked on as part of the BD2K effort is something called the BD bag, which is a Library of Congress specification to make data sets more portable. So we extended that to refer to big data. It's essentially a portable data archive system that lets you refer to data sets by their unique identifiers. So you can pass data by reference and kind of make the workflow systems a little bit agnostic of where the data is located. It's a lazy referencing mechanism where data is pulled by these lightweight API. So I mean, I was going to suggest if you could take a look at the work that we did in the BD bag effort. I can send you a pointer to that.
That'd be awesome. Yeah, I mean, that's definitely that's another level of abstraction we need, right? Is a standard way to do that. Yeah, it'd be cool to. I don't know if that's feeding in anything that the GA4GH is doing. I'm not into all the. There's so much work going on, and I know of it, but that would be awesome to be.
Yeah, we have a similar problem. We try to engage, and it's just too much bandwidth. There's so many things that you end up spending the day on telecons and meetings, and
yeah.
So I'll send you a pointer, and maybe we can have an offline discussion of how this could fit into the NIH Commons effort that we were all kind of funded on.
Cool. Yeah. And sorry, I know you had a second question, but I forgot when you were talking about that.
The second question is a parallelism.
Sorry. Yeah. So yeah, this parallelizes. So all the parallelism is defined in the common workflow language definition. So this will split by, like say for alignment, it will split by sample, and then within each sample, it can further split by reads in the fast queue if you want. and then then it comes back together, it gets merged, and then there's another parallelization step in, say, variant calling, where things that need to be called together get batched together. So like tumors and normals for cancer, or if you're pooled calling things together, they need to go together. And then each of those batches then gets split and can each run independently. And then within each batch, you can split by chromosome. It doesn't split by chromosomes, but chromosome regions, so like hunks of chromosomes. So yeah.
And that uses a construct within CWL to do the parallelism?
That's right, yeah. So CWL defines workflows and then sub-workflows within those. So
I see.
Yeah, so that's kind of the amazing magic that's happening, is that, yeah, you're just defining like, hey, this is a workflow, this is a workflow, and then each of the runners is taking that and then deciding how to parallelize it in the best way possible. So like Toil will just, like on a Slurm system, we'll just create a bunch of S-batch jobs and submit them, keeping track of sort of what parts of the workflow are waiting for other ones, and waiting to submit those until they happen. So yeah, and then DNA Access and Avatos and some bridges are doing similar things as well.
Thank you.
Cool.
Hey Brad, great to see you. Did you have to rewrite your workflows to CWL?
So we had to rewrite the way we define the workflows. So there was already sort of a workflow definition in bcbio, but it was bcbio specific, kind of Python.
Yeah, exactly. Yeah, for example, for Freebase, you had different options for different ploidy and different conditions. So does that have to be rewritten to a YAML kind stuff?
Yep. So we rewrote the sort of the definition workflow, but the actual workflows themselves are identical. So all the options that you had before now work the exact same on CWL. And so that's been now we have parallel bcbio with CWL and bcbio the old way. They both work under the same code base and the same set of things. What we like to do is not sometimes how to work, right, is to migrate everything. And so that's kind of what it is. But yeah, we don't have a separate set of workflows. It's the exact same biology underneath, just the workflow is different.
Okay, thanks.
Yeah.
Does CWL have a user, a graphical user interface?
Yeah.
Showing up for Photoshop?
Yeah, there's some nice ones. Yeah, I should show some actually. The Seven Bridges folks have what's it called? Composer. There you go. Thank you, Adrian's here to tell me. Yeah, this thing called Composer, which is awesome. It's like a graphical user interface where you can essentially like see the whole CWL, draw it up. You can select the options for each one, make it that way. That's by far the nicest one. Yeah. So yeah, I would definitely because otherwise it's writing kind of normal. Like in bcbio we generate it, so we don't handwrite it, but yeah, you can write it with pretty nicely.
Nice. Thank you. Brad, so if we're interested in running your favorite JTK variant calling pipeline on our system, we have the basic software installed and everything. But what infrastructure and what do we have to get from you and learn to be able to figure out how to work? Yeah, so
bcbio will install the tools separately. It kind of has its own, but I mean it's just a space really compared to like that much. But it's really there's a pretty simple install. It'll grab all that, it'll grab the data and everything, and then yeah, as long as you're running like I don't know if you're in SQ or what's cluster again.
Actually, we're on an LSF base.
LSF, yeah, LSF was fine with that. So yeah. And
okay. So as long as bcbio is installed on our system, then we could get to where you could share with us the pipeline, and then it would be trivial to run it.
Exactly. Yeah. Trivial. Yeah. Exactly. But yeah, it's just a command to submit and send it off. So yeah. Yeah, exactly. It should be easy. But yeah, that's exactly what it's trying to do. So that all the installs are automated. So yeah, and LSF should be no problem. So.
Great. All right.
Brad, again, are you validating against all the different platforms now?
I mean, kind of by hand, I guess, right? Yeah, I mean, all these are run by hand, but yeah. I mean, like the dream challenge is, well, you guys spin to it, so that's kind of trying to be like, hey, this is sort of a badge that you can run different places. And then I hope like the automation will be like, we can have some kind of monthly or something where it runs at different places, make sure it works, and we get back like, hey, it didn't work. Here's some that's the future.
Brad, I used the bcbio in last year.
Yeah.
But the project finished, so I haven't used it for a while. I have great fun using it. But looking at the slides, it seems that in the future, some comparing what we have. One thing I saw is the single cell variation, the audience will be.
Yeah. So I mean, Lorena or Mike are probably the best people.
Well, single cell RNA-seq in that case. So it would be single cell RNA-seq.
Oh, only RNA-seq, not variation.
Yeah. Oh, sorry. Yeah. RNC. Yeah.
Oh, okay.
Yeah.
Do you have something for single cell?
I haven't done that yet. I don't know. It's probably be interesting to see how it works. Yeah. Yeah. I mean, we have what we do have is a lot of UMI support. So if you have some kind of tagging scheme, it can definitely support that. But it doesn't like split by cells now. Call each independently. Yeah. The best way, I guess, would be to pre-split on UMIs and then run each independently, I guess. Yeah. I don't know. Yeah, there's not as many methods right now for that kind of thing.
So single cell coverage.
That's the problem, yeah. Yeah, that's what I'm not sure about it. So it'd be interesting to see. But yeah, I haven't ran it kind of. Cool. Thanks, guys. Thank you, everyone. Thank you, everyone online.
The schedule for the next big meeting in 2 weeks is open, actually, if anyone would like to speak then.